Hello. Today I want to continue my discussion about the structure of glomerulus. In the previous session you saw a schematic representation of the glomerulus here and one of its capillary loops here. This is a real picture of a capillary loop under the electron microscopy. You see here the endothelium with its nucleus here and the cytoplasmic extensions here, which you, as you can see, have some pores or fenestrations. As I told you before, the endothelium in the glomerulus is of fenestrated type. Here you have a mesangial cells, and these are the microfilaments, which is shown by MF here, microfilaments, which give the mesangial cell its contractile ability. This layer is GBM, which continues alongside the glomerular capillary loop, and also it covers the adjacent loops. Here is the lumen of the capillary in which blood flows, and these are food processes of podocyte or visceral epithelial cell, and this space is urinary space. Here you can see the filtration barrier through which filtration occurs under the electron microscopy with a high magnification. These are the cytoplasmic extensions of the endothelial cells, and these are pores or fenestrations. As you can see, these pores are rather large. This layer is glomerular basement membrane or GBM. As you see, GBM by itself consists of three different parts. This part and this part are lucent, and this part in the middle is dense. This part is known as lamina rara interna. This part is known as lamina rara externa. It means the internal lucent layer and the external lucent layer. And the middle layer is known as lamina densa or dense layer. As you see, GBM is continuous without any pores or holes inside it. These are food processes. You are familiar with them, food processes of the podocyte. And between these food processes, you can see a short distance, a short hole, which is named as slit pores. But this pore is covered by a thin layer known as a slit diaphragm, which is shown by the black arrow. slit diaphragm. So, if a substance wants to pass from the blood into the urinary space, here we have blood, this is the lumen of capillary, and this is urinary space. If substance wants to pass from blood here into the urinary space here, it should pass through these three layers: the endothelium, glomerular basement membrane, and the slit diaphragms. The endothelial cells have negative electric charge, and because of this they repel proteins. Most of them have also negative electric charge, so they are repelled by the endothelial cells. This is called as charge selectivity. It means that the endothelial cells can select the passage of substances according to their electric charge. If they have negative electric charge, they cannot pass through the endothelium, because endothelium has also negative electric charge. As I told you, GBM is continuous without any pores, and the pores between food processes are also covered with a slit diaphragm. Because of these two features, the continuity of GBM and the presence of slit diaphragms, large molecules cannot pass through these two barriers, and this is called as size selectivity. It means that GBM and food processes, along with their slit diaphragms, act as a barrier in front of passage of large molecules such as proteins. If for any reason the negative electric charge of endothelium is lost, or the continuity of GBM or the structure of slit diaphragm are impaired, charge selectivity and size selectivity of filtration barrier are also impaired respectively, and because of this, many substances such as proteins and albumin can pass freely without any problem through this barrier from the blood into the urine. And in these conditions, we have high amount of proteins in urine. This is a pathology condition, and it's known as proteinuria. Proteinuria means the presence of protein in the urine in high amounts, which can cause many clinical problems in the patient. So, the anatomical and physiological mechanism of proteinuria is loss of charge selectivity and size selectivity in the filtration barrier due to loss of negative electric charge of endothelial cells, loss of continuity of GBM, and the derangement in the structure of slit diaphragms. Thank you.